Many of you know that my laboratory works on trying to understand genetic pathways and pathophysiological mechanisms for human neurogenetic diseases. And we focus on developmental diseases. I'm a trained pediatrician and medical geneticist, and I'm sure that has some influence, but I think I got together doing development and those clinical specialties because of my overall interest in development, and I still do see patients. When I started my lab, we were exclusively using mouse models because at the time, a mouse was a great model system. It was a mammalian system where you could use ES cells and do manipulations of any genes that you wanted to and create models, a mammalian model, of a human genetic disorder and study the effects of that in a living, breathing, behaving organism. Of course, with being here and then at UCSF, I jumped on the human stem cell bandwagon because what's missing in mice is that there's some developmental processes and pathways that are different in the human. And also, of course, if one wants to do drug screening, as Evan alluded to, you really need to do it in human samples so that you can actually get drugs that work on the human proteins themselves. So we started doing this about 10 years ago. And so what I'm going to talk about today is our efforts to use both mouse and IPS models of a complex disorder called autism, which you all know about. And the complexity is sometimes daunting, but in using both of those models, a living, behaving organism and a human system, I think we can come up with some commonalities, at least in some forms of autism, that we hope provide some insight into this very terrible disorder. Well, the story that I'm going to tell starts with dishevelled, a gene family. And this actually preceded the time that I had my own lab. I was in Phil Leader's lab at Department of Genetics in Boston, and Norbert Perryman was cloning a number of developmentally important genes in the known developmental pathways in fruit flies. And one of the ones he cloned was dishevelled, or as Norbert would call it, if you know Norbert, dishevelled. It's not misspelled, actually. That's the way the gene is spelled, but you shouldn't spell it that way if you're writing any other manuscript. And dishevelled was part of, again, at that time, what we thought was a well-defined pathway of 5 components. So the ligand was known, the receptors weren't known, dishevelled, GSK3, and beta-catenin, and that was a well-defined pathway. And so our goal in starting this project was to see if we could use dishevelled, where we knew a pathway was involved, to look at developmental mechanisms with a pathway focus. So now, of course, the Wnt pathway involves many more than 5 genes, and this just gives an overview of 2 different pathways that dishevelled participates in. One of them is the canonical Wnt pathway, where in the absence of the Wnt ligand, GSK3 phosphorylates beta-catenin, causing its degradation of the proteasome. But in the presence of Wnt ligand, which binds to the co-receptors frizzled and LRP5/6, dishevelled recruits the destruction complex to the membrane, removing the GSK3 phosphorylation, beta-catenin accumulates, goes to the nucleus, changes transcription, and that then results in changes in cell fate specification and predominantly embryogenesis, what we study. There's another pathway that dishevelled is involved in, very similar in that Wnts bind to other frizzles. These are different Wnts. There are 20 different Wnt genes at least, and 12 frizzles. So a different combination of Wnts and frizzles results, again, in dishevelled, modulating intracellular pathways that now are important for cell polarity and migration and tissue patterning. So dishevelled plays this key role that we thought would be important. And so there were 3 dishevelled genes, and my colleague Dan Sussman, who was in Phil's lab at the time, cloned the 3 mouse genes. And we thought that if we were going to study dishevelled, it was likely they would have redundant functions. So we thought if we were going to study this family, we needed to knock out all 3 genes. So I'm going to take a little bit of a diversion for a minute and tell you about some of those studies. So being a logically oriented group, we knocked out 1, 2, and 3 and studied them in that order as single gene knockouts. And although there is redundant phenotypes, which I'll show you in a minute, we were fortunate that the single gene knockouts also had interesting phenotypes in their own right. And they were all different phenotypes. So for dishevelled 1, there was a unique social behavior phenotype that I'll tell you about in a few minutes. Dishevelled 2 knockouts in an inbred background, 100% of the mice had a somite abnormality that resulted in vertebral and rib abnormalities. But 50% of the mice in an inbred genetic background had a conotruncal defect. and only 2% had a spina bifida, a neural tube defect. So even in a completely genetically controlled environment, dishevelled 2 wasn't sufficient to cause a fully penetrant phenotype for some of these abnormalities. And then when we knocked out dishevelled 3, the single gene knockout, now 100% of the mice had conotruncal defects, and they also had a polarity defect in the inner ear. When we made the double knockouts, it revealed some of the redundancy. 1,3 double mutants, I'm also going to talk about those in a moment, had a slightly more severe phenotype that led us into studying parts of the brain development. But the 1,2 double knockouts now had a very severe neural tube defect with 100% penetrance, 100% penetrant conotruncal defect. Dishevelled 2,3 double mutants had axial abnormalities and the neural tube defects, as well as now also had inner ear defects as the dishevelled 3 mutants. And the triple mutants, we've just been studying now, and one of my graduate students, Justine No, is studying this in a little bit more detail, and the triple knockouts they fail to gastrulate. So this is also a collaboration with Hiroshi Hamada's group and Masakazu Hashimoto. Here's a day 7.5 mouse embryo. It's after implantation, and stained with brachyury shows the primitive streak and the developing mesoderm in this wild-type embryo. And the triple mutants, on the other hand, have no mesoderm and no brachyury staining, completely devoid of that. They have an axis. Here you can see what looks like maybe head folds, but totally undifferentiated. And so Justine's been studying these in embryonic stem cells, and for those who may be interested, I know I get a lot of requests for, do you have any triple knockout ES cells in general that one can study signaling? And it's been very difficult to make triple mutants in a single mating, and that's why it took us so long to find this phenotype. But we do now have triple knockout ES cells that Justine's made. And actually what's interesting is they maintain their ES cell character. They still don't make mesoderm, and Justine's used these cells to use an in vitro model to try to figure out more about the role in early embryonic development. But again, for anyone who's interested in a triple knockout, we now have such cells, and they're available once Justine publishes the paper. So just let me know if you're interested. So dishevelled is a multifunctional, redundant family. But I'm here to tell you about autism today. And how did we get into autism? Well, the reason we got into it was because of the dishevelled 1 mutants. And this was as I said, the first dishevelled that we studied. And so we really studied it in detail. Nardos Lijam and Rich Paler were looking in detail at adult animals, embryonic animals, and found absolutely no differences in any of the tissues, including the brains of these homozygous knockout mice. They seemed to have normal fertility, they had normal lifespan, they didn't get cancer, so everything was normal about them. We thought perhaps there'd be a brain abnormality, so we wanted to do behavioral testing. And so when we did behavioral testing of these mice, it's probably not the right thing to do, but I'm glad that we did it. We kept all of the mice in uniform genotype cages, so all wild-type genotype cages or homozygous knockout genotype cages. And in the old days, probably don't use this anymore, but we used these ear tags that had numbers on them so that we could differentiate each mouse before we tested them. And so every time the mouse was being studied for behavioral testing, they had to Nardos and Rich had to look at their faces and determine what their ear tag number was. And what happened after a period of time as they were doing this, they were saying, boy, there's something different with these mice. And they really couldn't figure it out, but then they did figure it out. And what was different was, here you can see that wild-type mice, most of them had no whiskers and facial hair, which was kind of surprising. But all of the dishevelled 1 knockout mice, caged as a uniform genotype, had full whiskers and facial hair. So we videotaped these mice, and I'd recommend mouse videotaping behavior for anybody that has insomnia, because it is quite. And then this is before there were a lot of more sophisticated tests, and we just tried to look to see what our gestalt was about their behavior, and it seemed like everything was normal, with the exception of their interaction with each other. So we looked in the literature, and it turns out there was one paper on social barbering. And an individual studied about 24 mouse strains and found that 3 mouse strains had social barbering. That is, put mice in a cage, the dominant mouse in that cage would either barber the other mice or allow the other mice to barber each other. And it happened in 3 out of about 24 mouse lines. And in general, that's an indication that that there's probably a genetic component to it as well. And so with that as a background, we tried to figure out tests that would allow us to determine whether or not this was a social behavior phenotype. So what Nardos and Rich did is they took about a dozen cages, and in each cage they took one mouse who was wild type and had no whiskers and facial hair, and one mouse that had full whiskers and facial hair and was disheveled, one knockout, and looked at their phenotype. And what happened was the disheveled mice all lost their whiskers, the wild type mice all grew their whiskers back in 2 weeks. Now put those mice back in their original cages, the wild type mice with full whiskers lose them again, and the disheveled one knockout gain them back again. Sort of the mouse whisker trimming crossover study for any geneticist in the audience. And so that, that's probably an abnormal behavior similar to biting your fingernails or picking your skin or things like that. But they actually did have some other abnormal behaviors that we saw in the videotape, and that was that they didn't sleep together huddled as wild type mice did, but slept together a little bit more disheveled, I guess you could say. And they didn't cooperate as a group to build these nice fluffy nests out of tissue paper wafers. So they seem to have real social behavior abnormalities. So we published the paper in the late 90s, and we thought it might be a model for a number of neuropsychiatric disorders, but this was the time when that was actually the first model of social behavior in any organism. So we were a little bit lost of what to do, and as I said, we had no brain abnormalities. But we thought autism might be a potential model, and so I'll just tell you how we got into that in just a second. But before I do, let me tell you a little bit about autism. As you all know, it's a group of neurodevelopmental disorders. Prevalence is rising over time and now is about 1 in 60 in the latest CDC incidence ratings. The core symptoms are the restricted repetitive behaviors, limited social interaction with communication difficulties. It's a heterogeneous disease, which makes it difficult to study. It's heterogeneous in its genetics. There's a number of environmental causes, and these probably interact. There's a difference in the severity of the core symptoms, and there's certainly variability in the comorbidities that are associated with autism. But one of those comorbidities was increased early brain overgrowth, macrocephaly. It occurs in about 20% of patients. And that was a phenotype that I learned about here at UCSD. Eric Courchain was one of the first to demonstrate that when looking at either head circumference or in MRIs of children that were at risk of autism, many of the autistic patients had increased brain size compared to normal, typically developing controls. And it happened in the first 3 years of life. And again, this is about 20% of patients. And at that point, there's several possible things that could happen to those children. One was the brains would go back to the normal size. In some cases, they'd continue to expand. In other cases, might even be macrocephalic or microcephalic at later time periods. Over the years then, Eric, and this was a 2011 study, actually took postmortem samples and counted the number of neurons that were in the prefrontal cortex. And what he found was there was an increase in neurons in patients that had early brain overgrowth and autism. And that's strong evidence that this is something that occurs prenatal. And there's a lot of evidence now that mid-gestation fetal life is the time where autism has its origins. And that then points to an abnormality in neurogenesis. And that being the case, we thought that this was something that we could study perhaps with hypothesis-driven experiments in the mouse and the human. And so what we tried to do was to use both our mouse model and induced pluripotent stem cell model of patients that had early brain overgrowth. Some of these studies, so Allison said that some of the work we do is well done. Well, that's because we collaborate with people like Allison. So we did work together on these cells along with Rusty Gage's lab, particularly Carol Marchetto. But the person in my lab that did the work and put both of these models together is Chaim Bellinson, and he's now a scientist at Teva Pharmaceuticals in Israel. So what Chaim wanted to do is he said, well, maybe the disheveled 1 mutants have no abnormalities, but maybe it's because of the redundancy. So let me look at some other compound mutants. And so he looked at the disheveled 1 homozygous, 3 heterozygous mice. And by this point now we have a little bit more of a sophisticated analysis of social behavior. And one of those things is to use the 3-chamber social approach. And this was developed by Jackie Crawley, one of my collaborators on the initial disheveled work. And what Jackie developed was 3 chambers where in one chamber you would have a wire cage with a mouse in it, and another you would have a wire cage that was either empty or had an object in it. You would then place a mouse in the middle of the cage where it had trap doors, open the doors, and give the mouse equal access to the other chambers. And what would happen is if the mouse was social, they would spend more time investigating the mouse than they would the object because they're interested in finding out if there's any threat from that mouse. And let's see if I can turn this on. In the movie itself, the top is a mouse in a 3-chambered approach with a mouse in the left side and with an object in the right side. And in this bottom one, there's a mouse with no object or mouse at all. So what happens? Well, the mice freely go to each chamber. The mouse explores the cup with the mouse much more than the mouse on the bottom, which is just randomly exploring the 3 chambers. And so if you just look at how a mouse behaves in the 3-chamber social approach with the mouse, you calculate how much time they spend with the mouse or the side with the object. And usually it's direct nose-to-nose interaction. And social mice will have statistically significant more time, spend significantly more time with the mouse than with the object. And both dishevelled 1 and 1,3 mutants are defective in that, and so they have social interaction abnormalities. And so then Heim said, well, based on the brain overgrowth model, maybe there's an embryonic time when the dishevelled mice might have differences. And so what Heim did was systematically look in all of the mutants at times throughout development from day 9.5 to birth, which is about 21 days. And what he found was, particularly at E14.5, the dishevelled 1,3 mutant mice, but none of the other genotypes, had an increased brain weight. And by the time they were at E16, and certainly at birth, that brain weight was now back to normal. So by sectioning those brains, and this is coronal sections through the mice, this would be the cortex up here, and this would be the ventral side, I think you can see that the cortical plate is enlarged, there's neocortical overgrowth in the wild type versus the mutant, the thickness is more. And this was associated with increased, with a change in Wnt signaling, a decrease in Wnt signaling, which one might predict. We were thinking this might be the case with the dishevelled mutants. And this is a top flash assay where we took neural progenitor cells from wild type and 1,3 mutants and just looked first of all to see what the Wnt signaling was in a luciferase assay. And there was a reduction of basal Wnt signaling in the double mutants. This could be corrected by a lithium chloride, which is a GSK3 inhibitor, downstream of dishevelled, but it could not be completely corrected with Wnt3A, which is upstream of dishevelled, suggesting that this was a dishevelled-mediated effect. That's in vitro. This is in vivo, looking again at E14.5 using a bat-gal model, which is a beta-galactosidase stimulated by Wnt signaling. And you can see that in the wild type, the bat-gal activity is high. And although the dishevelled mutants are somewhat variable, it was consistently reduced. And so it suggests that there's a difference, reduced Wnt signaling both in vivo and in vitro. Now, many of you know about mouse neocortical development, but for the next few slides, I'll just give you a brief summary. The progenitor cells in the mouse are predominantly the radial glial progenitor cells. And they stretch from the ventricular side, which is the apical surface, to the pial surface outside completely. And they divide symmetrically to make more progenitors. When they divide asymmetrically, they can either directly make neurons or mostly they produce these intermediate progenitors that are TBR2 positive. And when they make these intermediate progenitors, they then divide once or twice to make postmitotic neurons that migrate up the radioglia and then set up shop in an inside-out fashion as mature neurons. So the firstborn neurons are the deep layer neurons, and the later-born neurons become more superficial. That's how development occurs. So Heim then looked first in the ventricular zone to see if there was any increase or differences in the cell number there. And what he found was, again, only at embryonic day 14.5, there was an increase in the number of TBR2 positive intermediate progenitors and SOX2 TBR2 double positive progenitors. So there seemed to be an expansion of these intermediate progenitors in the mice. And looking more carefully then at E14.5, he used some of the deep layer markers such as CTIP2, which is expressed at 14.5, and there seemed to be an increase in the number of CTIP2 positive cells that will become eventually the deep layer neurons of layer 5 and some in layer 6. At later time points, at 16.5, the number of CTIP2 neurons now appears to be normalizing. And the number of more superficial neurons marked by CUX1 are in the proper place in the approximately normal numbers, although there could be a slight reduction. Fortunately for us, when we were here, we were doing a lot of collaborations with Jeff Rosenfeld, and so we had a marker for one of their transcription factors that marked layers 2, 3, and 5 in mice, and it was called brain2, a transcription factor, a POW domain transcription factor. So Chaim used that marker and found that layer 5 and layer 2, 3 were marked well in E16.5 brains in wild type, but in the double mutants, it was virtually gone, even though the layers 2, 3, and 5 are present in those mice, suggested that maybe brain2 was a downstream target of beta-catenin. So using neural progenitor cultures, first of all, Chaim found that the double mutant neural progenitors proliferated more rapidly, and they also had a reduction in brain2 in those cells. And so we collaborated with Jeff's group and got his brain2 knockout mice. And homozygous brain2 knockouts phenocopy the disheveled 1-3 phenotype. That is, thickening of the cortical plate, increased number of TBR2 positive cells, and increased production of the deep layer cortical neurons. And this is just quantified over here. But the homozygous mutants are embryonic lethal, so we use heterozygous mice and found that in fact they display social interaction deficits similar to disheveled knockout mice. So it seems that the brain2 mutant mice phenocopy the 1-3 mutants. And then overexpressing brain2 would rescue the phenotype in vitro by the proliferation phenotype. So how could we prove that this was in fact a embryonic phenotype? Well, we wished we had genetic tools to do this, but we didn't. And we still don't have them for the 1-3 double mutants. So Chaim decided to use CHEER 99021, which I will call CHEER from now on. It is a Chiron-developed, more specific GSK3 inhibitor to see if we could rescue the phenotype. And what Chaim decided to do was if he could inject pregnant females that would have wild type and disheveled mutant mice between embryonic day 9.5 and 14.5 daily with an appropriate dose of CHEER, could he rescue the brain overgrowth and the social behavior phenotype? And so when he did that experiment, the 1-3 mutant cortical expansion was rescued, looking at embryos. Another cohort treated in exactly the same way during fetal life and tested for social behavior abnormalities now had rescued social behavior abnormalities. So there's 2 points to make there. I think this really emphasizes that this phenotype, the adult social behavior phenotype, is likely caused by something that's happening in fetal life, very consistent with what we know from autism. But the second thing to point out for the future, and I'm not going to talk really much more about it now, it gives a proof of principle for how one might think about correcting such abnormalities during fetal life. And even though this is just a mouse, it's really only limited by technology and safety issues to consider how this might happen in humans. We're not doing any of that, just so you know. But I'm just saying it provides proof of principle. And in addition, we could then use MRI and fractional anisotropy to look at brain structures in the adult mice of disheveled 1 mock-treated and wild-type mock-treated mice to see what the brain abnormalities were different between those two. And using standard MRI, there were 21 out of 159 regions that we could quantify that were different in the disheveled 1 mutant mice. When we used fractional anisotropy to look at tracks, 68 of 159 regions that we could quantify were different. By we, this is Jason Lurch in Toronto doing this collaboratively. And then taking the disheveled 1 CHEER-treated mice with wild-type mock-treated mice, 6 of the 21 regions were now rescued. And what's important to note is that some of the regions that were abnormal between the two actually are regions that you would expect to be present from layer 5 abnormalities. There would be a reduction then in white and gray matter from layer 5, in particular primary motor cortex, somatosensory cortex, and the frontal association cortex. And these were rescued by treatment with CHEER in terms of their abnormalities. So again, suggesting that this phenotype that we're seeing in the mice actually is functional and is rescued with appropriate treatment. So from the mouse studies then, Heim uncovered an embryonic brain growth and social behavior pathway that included DVL, beta-catenin, and brain-2 and TBR2 that resulted then in an increased production of TBR2 intermediate progenitors at a particular time during development. And the idea would be, of course, that when dishevelled is normal, it would activate then beta-catenin. Beta-catenin would activate brain-2. Brain-2 would suppress TBR2, and you'd have a normal number of intermediate progenitors. But when dishevelled was lower, beta-catenin was lower, brain-2 was lower, and one would then have increased TBR2 since the suppression was relieved. And so that seems like a transcriptional cascade that's working in the mouse. But of course, any of you have seen a mouse brain and a human brain. I don't think this is exactly to scale, but there's many differences. Included in that, besides the size, is the complex structure of the human brain, including the gyri and sulci. And of course, there are also an increase in the proportion of various progenitor pools. In contrast to the mouse, where I told you the development was predominantly radioglia driven, in the human, there's a new class of progenitors, the outer subventricular zone radioglial progenitors, that are not attached to the apical surface, but produce a number of the neurons during mid-gestation in the human brain. And these are present in the mouse, but a very, very low number. So really, one can only study these in the human brain. Now, we don't, other than people in California, and maybe not anymore as well, we don't really have access to human fetal tissue to study this. It has been studied by Arnold Krickstein, among others, and so this has been confirmed in human tissue. So what can we do? Well, of course, the option that we have, which is what makes studying the brain with induced pluripotent stem cell technology so appealing, is that we can make such models from human diseased individuals. And of course, you all know this technology, which was developed by Shinya Yamanaka now 15 years ago. Skin cells or other cells from a patient can be established in culture, reprogrammed into pluripotent cells, induced pluripotent stem cells that are just like ES cells for all intents and purposes. They can make a particular disease type for a disease model or test it for cell therapy. We're fortunate too that iPS cells in general like to be made into neurons and neural tissue, so it makes it easy to use for models of human neurogenetic diseases. So when I was still at UCSD, Rusty and Allison and I collaborated with Eric Corshane, who had a number of patients that had early brain overgrowth. And you can't really read this, but this is from the paper. Each of the individuals had autism by testing. They all had early brain enlargement. And so if you looked at the MRIs from all of these samples, the brains on average were much larger in the autism samples than the control. So 8 lines were established from autistic patients with early brain overgrowth and 5 controls. They were named cleverly with C for controls, with different words. And some people said, why do you use this lab jargon? Well, actually, it's just something that's actually pretty convenient. And all the autism lines start with an A, so you guys can use the lab jargon to keep straight. I still do have trouble, though, saying, which is the cell line that has this phenotype? Anyway, so what we were interested in, Allison has been interested in these and has done direct neural differentiation, has found that these cells have very abnormal networks from using multi-electrode arrays. And Rusty's lab with Chris Glass has just recently published a beautiful epigenetic study. But what we decided to do was focus on the NPC phenotype. So we took the AST lines and the control lines that, again, were made into IPSs and made neural progenitor cells. And then simply asked the question, did the cells proliferate differently? And to our surprise, but again, it was hypothesis driven, so we were glad that we had this hypothesis. This is doubling time of the cells. So a larger number means a slower time of proliferation. All of the control lines, this is the average value, control lines are proliferating slower than the autism lines. And this is the average number, but if you take each of them individually, each of the autism lines proliferated faster than any of the individual control lines. So there's a clear proliferation phenotype, suggesting that at least in the patients that we're looking at, this early brain overgrown phenotype is associated with IPS model NPCs that proliferate faster. Okay? Well, then we wanted to see if they were similar to the dishevelled model, and to our surprise again, we found that in fact the autism lines, the 8 that we were studying, all had a decrease in basal canonical Wnt activity using the top flash assay. Now, trying to rescue it with lithium chloride or cheer, this wouldn't rescue. So it's not a problem with dishevelled, it's some overall abnormality with Wnt signaling that we're not clear about what that is at the moment. And the same thing was true for upstream using a ligand, Wnt3a. So there seemed to be abnormalities in beta-catenin. There were also abnormalities in brain 2. Just like the dishevelled knockout mice, brain 2 staining of the neural progenitor cells derived from the control were obviously a relatively high level, and in the neural progenitors from the autism lines, that staining was reduced. TBR2 was not expressed in these two-dimensional cultures that we're talking about. We'll come back to that a little bit later. And overexpression of brain 2, shown here by the stained cells here, would also rescued the proliferation defect. So here's the untreated cells, or treated, transfected with empty vector. The autism cells proliferate faster, but if you overexpressed brain 2 in those cells, now they proliferate at the same rate. So again, suggesting that it's a brain 2, now it's a beta-catenin brain 2 phenotype that's responsible for proliferation in this human model of autism. So there's some really interesting similarities here. And so we've tried over the last few years to try to expand those studies, and included in that, we're doing some mouse genetic studies. First of all, we have a number of different back alleles where we can modify dishevelled to focus on either disrupting the canonical Wnt or the PCP, the Wnt PCP pathway. And we're then trying to see if any of those rescue the social behavior phenotypes and the brain overgrowth defects. The second genetic studies, we're trying to see whether dishevelled 2 is involved. Because dishevelled 2, we have a conditional allele, and we'll be able to be a little bit more have temporal control over when we knock out dishevelled 2. And it turns out from these experiments that, I don't have time to tell you about them today, but we don't really have any from the allelic series that we have for the dishevelled 2 alleles, all of them seem to rescue the social behavior phenotype. And that might suggest there's a role for overexpressing any of the disheveleds and correcting this phenotype. But also that dishevelled 2 is involved in social phenotype as well in the 1-2 double mutant. So we're trying to study this a little bit more. We're doing genome editing to correct mutations that we found in the ASD lines. We've done genome-wide RNA-seq and ChIP-seq studies to look for genes and pathways regulated by beta-catenin, brain 2, and TBR2. And I don't have time to tell you about that right now, but we're collaborating with Rusty's lab. Again, as I told you, they did a nice epigenetic study and did ATAC-seq on all of these lines. So we're merging our RNA-seq, ChIP-seq with the ATAC-seq. And in fact, we've done cheer rescue of these lines, slowing their proliferation. And we're looking to see what pathways are rescued, and those experiments are in progress because we just got the data from Rusty's lab now. As I said, by using the human lines, one can do drug and small molecule screening, and we're just trying to see if there are any other pathways that are disrupted here, because even though we found a Wnt beta-catenin brain 2 TBR2 pathway, we don't think this is the only thing that's going on here, so we want to see if there are other drugs as well, and we're in the process of doing that. I really have nothing to say now. And we're using three-dimensional organoid models to study brain development, as I'm sure many of you are doing here. So in the interest of time, I really only have time to talk about the genome editing and a little bit of the organoids work that we're doing to follow up on these studies. Well, the first thing I neglected to say so far was that we actually found some interesting mutations in 3 of the 8 lines of the autistic individuals. One of the lines, ARCH, has a loss of function mutation in beta-catenin. And it's at amino acid 73, which is in the 3rd exon. It's a very early mutation, so we think it's likely to be a null mutation. And this has been seen in other autistic individuals, but mostly ones that have microcephalic phenotype and intellectual disability. So this individual has this heterozygous loss of function mutation in beta-catenin. Another individual for Asai has a early loss of function mutation in frizzled 6, and that's at this, I'm sorry, 116, I think, amino acid. And that, frizzleds are 7 transmembrane G-coupled protein-like receptors. And so this mutation is in the extracellular domain, so unlikely to make a receptor. It might make something that interferes with Wnt signaling at some level. We don't know that. However, most likely it's also a null allele due to nonsense-mediated decay. So we have 2 lines, each of which has a mutation in canonical Wnt pathway members. And the third line is APEX. APEX has a missense mutation in PTEN. And the missense mutation is in 130, amino acid 135. I'm sorry, I have a little bit of trouble on the side way looking at this. And all of you are aware of PTEN's important role in growth proliferation. And what PTEN does is work in the PI3 kinase pathway to suppress the activity of AKT and then increase apoptosis and decrease cell survival and proliferation. It's a well-known pathway in cancer biology, but of course, mutations in individuals with autism and early brain overgrowth, about 5 to 6% of them have mutations in PTEN. Now, what's interesting is most of the autism-derived mutations in PTEN are milder mutations. That is, if you have a complete loss of function mutation of PTEN, you generally have a cancer-prone phenotype like Cowden syndrome. But milder mutations, and these have all been assayed in yeast and other systems, those are the ones that appear to be more associated with autism. So that would be consistent with this finding here. So we have these very interesting mutations. Of course, when you then have a reduction in PTEN activity, then you increase cell proliferation and decrease apoptosis. So the question then that we wanted to ask is, can we use these lines to see if the Wnt pathway and PTEN pathway play a predominant major role in regulating the proliferation? And so Shuai Fu, a graduate student in the lab, has been doing the following sets of experiments. I know this is going to go forward, so I'm just going to put them all out there now rather than... So what Shuai's done is in each of the lines here, so these are the 3 lines that have the mutations in beta-catenin, frizzled 6, and PTEN. And our control lines here in green are wild type in each of those loci. So what Shuai's doing is taking the Cas9 nickase activity, so it only makes single-strand breaks surrounding the mutation sites in each of these, for each of these 3 mutations in these lines. With the appropriate guide RNAs. And then using homologous recombination, correcting the mutation to the wild type residue, or in the control lines, introduce the mutation into the control lines into the same spot. And so that has a couple of advantages doing it 2 ways. First of all, we're going to just test in an isogenic system. So in this line, we have 2 possibilities then. We'll have the mutation isogenic lines in the background of autism, or in the control organism, we'll have the control and the mutant line. So is it the background, is it the mutation itself, or is it a combination of the 2? So Shuai's had some success in a number of these ones. I'm going to tell you about one of the experiments now because he had an unexpected result that came from his targeting. So as you know, one can often have larger effects when you're trying to be very specific in making some of these modifications. And what Shuai found, he tried to introduce in one of the control lines, CHAP, was introducing the mutation. And he found that one of his lines had a large deletion. When he did the sequencing, it was obvious that he had this large deletion in this region here that would likely then be a loss of function for PTEN. And he found one that was heterozygous and one that was actually homozygous. And here's the proof that these 2 clones are missing PTEN. This is the PTEN protein with 2 different antibodies. And in 2 of the lines, he seems to have a homozygous knockout of PTEN itself in these lines. So he looked first then of whether there was a difference in proliferation. And this is preliminary data. He used ARCH rather than the line APEX that has the PTEN mutation. But ARCH itself proliferates rapidly. CHAP, the line that the mutation was introduced in, proliferates more slowly, again in passages 5, 6, and 7, just like we saw earlier. But introduction of PTEN knockout, now there's more rapid proliferation. So this mutation seems to have an effect in the control line. He's also, though, been able to introduce the PTEN mutation in the same line, CHAP. Here's the heterozygous line, And here's Apex with the heterozygous mutation compared to the CHAP control. And so he's now introduced the heterozygous mutation, and what happens when he does the proliferation studies as well? Well, again, looking at CHAP, the P10 knockout, or the P10 induction line with just that point mutation, both of those P10 mutant lines now proliferate more rapidly than the control line. And they're similar in proliferation to the arch line that has a beta-catenin mutation. So this would suggest that these P10 for sure would be a major effect. We're now testing whether the beta-catenin and FRZLD6 mutations have similarly important and dominant functions. So finally, I wanted to end with some of the studies that we're doing with organoids. And I'm sure, as I said, many of you are using IPS models for brain studies or using organoids. And this is a study from Yoshiki Sasai's lab, which is the protocol that we generally use because it's focused on the cerebral cortex. And here's the general protocol. I'm not going to go through it in detail in the interest of time. But two of the people in my lab who pioneered, let me just show for those of you who don't use organoids, that one makes organoids from the IPS cells, and the organoids themselves are just balls of multiple ventricular zone containing structures. And these structures contain proliferating zones that looks like the ventricular zone of a mouse. It has an apical polarity, just like there would be in a normal human or mouse brain with the apical surface. They produce neurons that are in the cortical plate outside of the ventricular zone, using several different markers of stable neurons. So it seems like a great system. And so two postdocs in my lab, first Marina Burstein when I was at UCSF, developed this using a lissencephaly model, and Luke Burry in my lab is doing the experiments with the autism lines. And so I'll just again point out that in these organoids, when you look at detail at the ventricular surfaces, you have all the proliferative NPCs that we would like to study. So certainly we have the radioglial progenitors, intermediate progenitors, TBR2 positive cells. They make neurons as shown by CTIP2 up here, and they make the outer subventricular zone markers marked by HOPX here in these organoids. So we can then study the role of autism and other mutations in each of these progenitor populations, including HOPX positive ORGs that can't be studied in the mouse really. And so here's what the organoids look like. This is in a model of microcephaly, which was a lissencephaly line here, Miller-Dieker syndrome. And one of the things that's interesting about organoids is that you can use them generally for proliferative phenotypes. So if you study microcephaly, almost all microcephalic lines in models are smaller than a typically developing or normal line. And we've tried to do this with the autism lines, but we really haven't had any consistent phenotypes. And I think it's because there are multiple ventricular zones within those organoids, so it's not been consistent, but we're still trying. But for microcephaly, it's quite good. And here's some examples of some of Marina's experiments where she's looked at, again, the Miller-Dieker lines versus wild type. And here you can see that the ventricular zone is very well formed with proliferating cells marked by Ki67. The white cells are SOX2 positive cells, reduced in the microcephalic lines, with at least in some of the lines there's apoptosis. So you can really see a mechanism for the microcephaly here. And if you zoom in on the ventricular zones here, I would challenge anybody in the audience, I can tell you they're from organoids, but I challenge anybody in the audience, if I didn't tell you this, to say that this wasn't from a normal developing human or a normal developing mouse, because it has very good formed apical surface. Here's the DAPI staining to show all the nuclei in the ventricular zone. And not only that, the cells divide, undergo S phase right at the ventricular surface, the apical surface. And here are cells marked by phosphohistone H3, which are undergoing mitosis. And they're right at the apical surface as you would expect. And so you can even use these to look at angle of division or anything else for the properties here as well. But you can also, of course, do live imaging studies, and this is one of Marina's studies, to show that in fact we get the behavior of the ORG population of cells. So going back for a minute, the ventricular zone radioglial cells divide at the apical surface, and they divide with a vertical sort of cleavage plane. On the other hand, ORG divide in a horizontal plane, and they don't attach to the apical surface. So as I show this movie, I want you to look at this, I think it's this cell right here. You have to look quickly because it jumps pretty well. So that cell's going to divide, and I'll tell you what it's going to do first. it will jump up, divide horizontally, and then stay there for a few minutes so you can marvel in what the cell did. So it jumps up, divides horizontally, and now moves away from each other, just as a typical ORG performs in live fetal brains as well. And you can show that that's an ORG cell by using markers specific for ORG cells after you finish the movies there. So this is Marina's study, and it was beautiful. And she used that to show that in the lissencephaly models that we have, there's a problem with the division of the ORG cells. I'm not going to talk about that now. And for those of you who didn't see that quick movie, there's a jump up, horizontal division, and then that's how they divide. Well, Luke's used these models, and he started to quantify some of the cells. Quantification in the organoids are a little bit difficult because there's a lot of heterogeneity, as anybody who's used them are aware. But what Luke did was just do a simple experiment that obviously takes him a long time. It's simple in concept, but difficult in execution. That is to take multiple organoids, multiple tissue sections from those organoids, and try to determine in the ventricular zone what the ratio of TBR2 to PAX6 cells are. PAX6 would mark the radioglial progenitors. So in the CHAP and the COVE control lines, here's the level of that ratio here. Two of the lines, ARCH and ASI, these are the ones that contain the Wnt pathway mutations, both have what appears to be an increased number of TBR2 positive cells. But APEX and AHOY, APEX has the PTEN mutation. We're not seeing that change in TBR2. So this would suggest that there's a conservation in these two Wnt pathway mutation autism lines of this beta-catenin brain to TBR2 connection. Although we're not sure that whether it's going to be present in the other lines as well. So again, we're using organoids. They resemble early cortical development, different types of progenitors present, and they're localized properly. There's appropriate production and timing of neuropopulations, and the ORG cells are present. Future, we're going to, as I said, try to look at size, proliferation rate of these progenitor subtypes, gene expression profiles, and we're going to do sulfate labeling to try to more accurately quantify the HOPX or positive ORG and TBR2 positive IP intermediate progenitor cells. I'll just show you how we're going to try to do this, just for your interest. We don't really have any data to present about this, but what Shuai Fu and Luke Burry have done is to try to use a system in which they take and introduce this fusion gene, which would be a broadly expressing CAG promoter and a turbo RFP that's MIC tagged that has a stop floxed cassette in there so it's not active, and inserted that into one of the safe harbor sites at chromosome 19. And then in a variety of different lines, they're taking CRE that's driven by the TBR2 promoter, but in such a way that TBR2 isn't going to be influenced, so it doesn't have any influence itself. So it's introduced here with the appropriate markers. So when CRE then is produced, it'll remove the stop site, and then any cell that originated from a TBR2 positive cell will be labeled in red. Same token, and so one can look at all the neurons that are made from TBR2 positive cells. Using the same sort of strategy in chromosome 22 safe harbor site, they're putting in a ZS green flag tagged cassette, also driven by a broadly active promoter that also is stopped by the stop flox. And this is now, rather than flox, it's with a ROX sequence. So they're introducing then DRE into the HOPX promoter in the same way so that HOPX expression is maintained. And then when you relieve, when HOPX is expressed, those cells will be labeled in green. And so what they're trying to do then is to put these into each of the individual autism and control lines, as well as in the genome edited lines. And they'll be able to quantify then number of cells that are coming from those and whether those cells are going to be different, as they often are from different progenitor pools. Likely they're different progenitor pools because the output is going to be different. So we'll have a tool then to use these. Now, I wish I could tell you that it's all working right now. It's not working right now because when we introduced those genes into the safe harbor sites, they were silenced. So we're putting them in again as with insulators, and I think that's going to work that way. But in any case, hopefully at some time in the near future, if I ever come back, hopefully not 10 years later, that I can tell you about those studies as well. So in summary, I've told you about conserved neuroprogenitor phenotype in both human and mice that have disrupted Wnt brain2 TBR2 pathway, the DVL1-3 mice, and patient-derived neural progenitors. And there are lots of similarities between the two. And we're going to try to use those similarities and differences to see what the key features are in the neural progenitor phenotypes, as well as hopefully have some insights into autism. And we have a lot of collaborators. I've told you the people in my lab, for the most part. As I said, Chaim Bellenson is the main driver, but Justine, Chen, Luke, Wendy, and Shuai also use their data as well. Jeff Rosenfeld, Nadav Ayatub from UCSF, and Jason Lert from Toronto I mentioned. The IPS work was a collaboration with Rusty's lab and Allison's lab. Again, I want to point out Carol, here's funding. Something else is over there as well. Oh, and Eric Corshane, who had the initial patients. And I thank you very much, and thanks for letting me come back to San Diego.
Hi, I'm wondering, in your dishevelled mice or the ASD iPS cell culture, do you see any defects in other cell types rather than the excitatory neuron lineage?
So in the brains of the dishevelled 1-3 mice, we haven't seen anything that's different. And of course, we look carefully at the dishevelled 1 mice over many years. I'm sure there are other abnormalities there. I think that one of the things that's really promising about the MRI studies is that it can reveal some of the organizational differences in the adult brain. So I think we can go back and study the different populations of cells and how they might interact. So I'm sure there's some differences at that level. But at the moment, other than the ones I've described, we haven't found anything different. And by the same token, at the moment, we haven't found any differences in the organoids other than the markers that I showed you.
Great talk here. Regarding the mechanism of the dishevelled function, I wonder if you look at a SOX2 activity, because the Wnt signaling beta-catenin in particular has to evoke SOX2, as Rusty showed, from NeuroD and other neural promoters. And also brain2 has been shown to cooperate with SOX2 neuroprogenitor. So it was consistent with the increased activity, or I should say, increased proliferation, of neuroprogenitors that will decrease SOX2 activity, So did you check SOX2 activity?
Well, we use that as a marker. So we use SOX2 or PAX6 as markers of those cells. We don't really see any quantitative differences in immunohistochemistry, but we haven't done any more quantitative studies.
Right, because the level of SOX2 wouldn't predict it to be changed. It's the activity.
Yeah, I didn't. So didn't see any change in the level of staining or the number of cells that were there in the SOX2 positive.
I was curious as to whether it looks like you have an early increase in brain weight of the defective mice relative to normal fetus. Is that also seen in humans? Is that accompanied by an increase in intracranial pressure?
So in humans, it's difficult to study because you can't identify fetal autistic individuals for the most part unless it's a single gene mutation. But most of the brain weight changes occur postnatally in humans, because even though there are more cells, the weight occurs because of the elaboration of the arbors and things of that sort postnatally. But that doesn't result in increased intracranial pressure. The brain is increased in size, the skull accommodates that, and there doesn't seem to be generally a problem with increased pressure.
Do you also see the same kind of proliferation in spinal cord, or is this only a cortical phenomenon?
We haven't looked in the spinal cord. It's a very interesting point, but we haven't looked there as well.
Yeah. As you know, some of the kids who have big brains have an enlarged glial population. Could you comment a little bit on what have been going on with the astrocytes of the glia and a lot of what you've observed? We
haven't looked at that in the organoids because we don't really take them long enough to have development of any of the glial populations. And in general, a lot of the studies there have been done later on in life in individuals that have autism. And so whether it's a primary effect or something that's secondary to having too many neurons during early development is unclear, because it's hard to look at these brains prior to the diagnosis of autism. The diagnosis usually occurs at 2 or 3 years of age, so who knows what's happening in those earlier time points.
And even in the mice that you reported early on,
we don't really see any differences in the glial populations in the mice. Yeah, we don't really see that.
Thank you again for your talk. I wanted to get some more information about your drug screening effort to reduce NPC proliferation.
Yes.
You mentioned previously that cheer, as you say, could actually rescue some of the social phenotypes that you noticed. Were you able to actually couple lithium chloride and CHIR, and specifically the ACI cell line that actually has a mutation in Wnt?
So we designed the experiment to look at a couple of different autism lines versus a couple of different control lines as our primary screen. And the ones that we used were, I believe it was one of the ones with the Wnt mutation. I don't remember, it was the beta-catenin. I think it was more likely to be the frizzled, because we wanted to make sure we could impact downstream of that, and we didn't want to mess with the beta-catenin. And then we used a line that didn't have any known mutations. And the primary screen was if those two lines had their proliferation rescued without an effect on two of the control lines, then we went further to screen all the different lines. And we've come up with a few different compounds. And we haven't done anything further than this at the moment, but they're promising compounds. Some of them one might expect, some we didn't expect. And so Chen in the lab is going further on that now. I don't really have anything else to report. The next step is if we do find those, we'll use them on the dishevelled model to see if they rescue that phenotype as well.
So if any other questions, let's thank Tony for the wonderful seminar.
Thank you.